All right, good afternoon. Pleasure to have you all back after lunch. Hopefully the postprandial levels of glucose will maintain you guys awake, and we'll make sure we do our best up here. So I'm Devyn Smith, the CEO at Arbor Biotechnologies. We are a gene editing company. I'm pleased to have three esteemed gene editing experts up here, and what we thought we'd do today is just start by just having each of them introduce themselves, a little bit about their role in their company. So we'll start with Alex Harding.
Hi everyone, I'm Alex Harding. I'm head of business development at CRISPR Therapeutics, and just joined the company at the beginning of the year. It's been a very exciting time actually for CRISPR. I'm flying back after this because tomorrow is the 10th anniversary of CRISPR as a company. So there's that milestone. And then October 31st, Halloween, we have an AdCom for exa-cel. And then finally, December 8th of this year, PDUFA date for what could be the first approved gene editing product. And so we're very excited about that as well. So a lot going on in the company. Also really excited to talk about what's coming next beyond exa-cel, next generation of gene editing that we're working a lot on.
Great, thank you very much. Hari.
Thank you, Devyn. Thank you for the invite, and thank you all for showing up here after lunch. My name is Hari Pujar. I'm the Chief Operating Officer of Tessera Therapeutics. Tessera is a privately owned biotech company in the Boston area. We're a Series C company. We're focusing on gene writing, built on the platform of mobile genetic elements. So we do writing and rewriting of the genome. We're about 350 plus people, headed by Mike Severino, who came over as CEO from AbbVie, where he was a vice chair. We're excited to add several more tools to the toolbox of gene editing, and you'll be hearing about all of those in upcoming months and quarters.
Great, thank you. And last but not least, Laura.
All right, so I'm Laura Sepp-Lorenzino. I'm the Chief Scientific Officer of Intellia Therapeutics. Intellia is a clinical stage company doing CRISPR, both in vivo and ex vivo. We are building a deep and broad toolbox of editing and delivery modalities. There is no single editing tool that will allow you to do all the therapeutic edits that you may want to do. So we've invested and continued to do so in having a couple of different enzymes, Cas enzymes that we can use as cleavases in the canonical mechanism, as well as base editor and a DNA writer. And it's important to deliver them. So we are also really working hard, not only on our LNP, which is clinically de-risked, but also pushing to see where LNPs can go out of the liver, as well as AAVs. And looking forward to this discussion. Thank you.
Thank you, each of you. So as we look ahead, I think Alex laid out a few of the milestones from CRISPR. There's a lot of, I think, exciting clinical readouts from Intellia over the coming year. What do you see, each of you, as you look out, say, the next 12 to 24 months, what are the biggest opportunities and the biggest challenges you see facing sort of gene editing companies as a whole? And we'll start on that side, Laura.
So look, 24 months or 12 months from now, Alex, I do hope we're going to have the first approval with exa-cel. It's really, really exciting, and it's going to be an important milestone for CRISPR Therapeutics, but for the whole field. So I'm really, really excited about that. For Intellia, we have two programs with very strong clinical proof of concept that are moving to phase 3 pivotal trials. So that is going to be very exciting for that. Both of them were using a knockout approach, but we're also filing an IND CTA later this year for our first insertion program. So it's continuing to push CRISPR in its different modalities to allow us to do that. We are also very excited about working with a number of collaborators. We are partnered with Regenerative. We recently expanded our partnership with them, looking at targeted AAVs, so getting out of the liver. So we believe that that is imperative, as we want to realize the value of gene editing. And then there is going to be all these tools, right, that we need to realize and put into motion by ourselves and through partnerships. I'm excited about we have an allogeneic platform that leverages our multiplex editing using a base editor and a CRISPR insertion, as well as the DNA writer, and the versatility of that tool as compared to other tools.
Very good. All right.
At Tessera, as I mentioned, we're developing a suite of tools for gene writing and rewriting, primarily using all RNA as a composition of matter. So over the next 12 to 24 months, we're looking forward to bringing these technologies to bear across multiple therapeutic areas, whether it be rare genetic diseases, or sickle cell disease, which is rare but one of the largest ones, oncology. But the idea ultimately being move the field forward, from ex vivo to perhaps fully in vivo type applications, and also have tools that don't rely on double-strand breaks. So that's what we're looking forward to. But kind of taking a zoomed out view, it's just amazing to see the field mature in a manner where the time between discovery of a new mechanism for gene editing and its application in the clinic is constantly shrinking. And that's very exciting, and that just goes to show the maturity of the overall field, and that's great for patients. And so I'm excited to see how we advance as a field. There was a statistic about 60% of approved medicines in 22 have a genetic basis, and as we look forward 12, 24 months, I think that percentage is likely to increase. And so we're all in the right place.
Yep. Hari and Laura both touched on it. There's a sort of mutual benefit in this field. As one company advances, the other companies benefit from the experiences that company has had. In our case, with exa-cel getting hopefully to approval by the end of this year, there's benefit, there's tailwinds for other companies. Likewise, with in vivo gene editing, getting into the clinic and getting more data there's kind of reciprocal benefit for us. And so one of the things that we're looking forward to at CRISPR is getting our in vivo gene editing programs into the clinic. We've disclosed that we'll have our first in vivo program into the clinic this year. That's ANGPTL3, which is for cardiovascular disease, and it's using a lipid nanoparticle targeting hepatocytes. Beyond that, we have a program for Lp(a) that's going to get into the clinic in the first half of next year. And then there's really a large number of other in vivo hepatocyte-directed programs beyond that, and then you can think about going outside the liver or doing other types of edits besides knockouts.
That's good. So, a lot of good things to look forward to. Hopefully a lot of good tailwinds over the next 12 to 24 months. You alluded to something, Alex, which I think is interesting, is, there's right now most of the editing programs in vivo are liver-centric. And how do we get outside of the liver, and how do we get outside of sort of knockdowns? Because that's sort of where the concentration, which makes sense, it's the technology, it's the starting point. How do we move beyond that? And be interested in sort of perspectives across the board on this. We'll start with you, though, Alex.
Yeah, we're working on it. As it sounds like my other colleagues up here are working on it as well. We have an LNP group internally at CRISPR. We've put a lot of resources into investments in that area, and that's really focused on LNPs for delivery outside of the liver. For us, an area of interest certainly is hematopoietic stem cells. We have exa-cel as our lead program, which is an ex vivo treatment that requires conditioning therapy. And so if we could get to an in vivo editing of hematopoietic stem cells, you could obviate the need for the conditioning regimen, which would open up that treatment to a large number of patients. So that's an area of high interest, but also LNPs to other tissues and other cell types is of interest. And then, we're open to other modalities beyond that, whether it be non-viral or viral modalities, where there's an opportunity with a specific application that we need to address, then we look for the technology that would allow us to deliver to that tissue.
Good. Anything to add, Ari?
Yeah, and as Alex mentioned, there are two components. Alex talked about delivery, and certainly at Tessera, we have a major focus on delivery to extrahepatic tissues. In fact, we presented some data at JP Morgan that has continued to evolve even further for delivery to hematopoietic stem cells and T cells. And in some ways, after the liver, the tissues that are accessible are the blood cell types. Lung is another one that's actively being worked on. And so I think we're moving beyond the liver. But I think we still have a long way to go even in the liver. Delivery of LNPs to the liver beyond a certain dose is still a challenge. And so I think delivery as a field has a long way to go from where we are today. Dosing more than 1 mpk is still a challenge for the liver. And as you go beyond the liver, you'd have to worry about dose, et cetera. On the editing side, as we presented previously, we have tools, we call it RNA rewriting, that can precisely insert, replace, or delete small sequences or add whole gene sequences into the genome. And so those are the ones that we look forward to bringing to the clinic going forward.
So Harry, I think you need to go look at our clinical data on LNP. You can have very safe and efficacious LNPs in the liver that you can get to 98% editing in every cell with pristine, clear, safety. So it's not just having an LNP; it's making sure that what you have is meets your target product profile and exceeds it. So with regards to to getting out of the liver, right, is, we do have a collaboration with Sparing Vision that's allowing us to, we have a small cast that fits nicely on an AAV, so it allows us to optimize that. And some of those lessons learned now we're going to be applying to our work with Regenerative and using their targeted AAVs for muscle and CNS. And then with regards to LNPs, there is an enormous opportunity on taking them and applying them, optimizing for, the organs, the tissues where they're going to work. And there is going to be certain tissues where, these are particles, right? So they will not get, to a certain number of tissues. So understanding that and, and it's through the application of, working with the surface characteristics and the protein corona and or targeting, right, that you can maximize this. But the advantages on the delivery is needs to be a good merge of editing and delivery, right? So for example, it is key that whether you're using an LNP or an AAV, that the expression at least of cases that are bacterial in origin or from bugs, that needs to be transient because you want to minimize the risk of immunogenicity. So that's, if you're going to work with AAVs, keep that in mind. Having constitutive expression of your editor is not a good idea.
If I may just follow up, I congratulate you on excellent clinical data that you've published with the liver LNPs, and by no means I was commenting on that, but my point was that we're still in the early innings of even liver LNPs. That if you wanted to deliver a much higher dose, for example, one runs into...
Why would you need to do that if you're getting to 99% of the cells?
For the kinds of knockdown approaches, you're absolutely right. But I'm talking more broadly in terms of delivery to the liver. And of course,
one gap we have there is with DNA delivery to the liver,
exactly.
right? Where it's not so much about the dose maybe, but it's about the immunogenicity of the cargo. And that's an issue that's not been solved with the non-viral approach.
Exactly, and if you wanted to get into the nucleus, for example, I mean, having to deal with the innate immune system in the cytoplasm with the DNA and nuclear entry. So these are still many problems to be solved in our field.
I think delivery is an interesting piece of the puzzle. Obviously, the moving out of the liver. There's non-viral. There's viral approaches. There's I don't know how many companies out working on delivery now, but there's a lot of them. And do you think we're going to have one platform or one approach that does it all?
No.
Definitely not, right? So I think the way we take our approach is start with the application, the end application. What is the disease that we're trying to treat? What's the edit? that we're trying to affect, and then go back from there. So what cell type is that in and what tissue, and then look at what delivery methods might be most useful for that.
Couldn't agree more.
So, you know, so one of the other pieces that I think obviously we hear a lot, you know, from investors and others is always around safety, right? And off targets, and, you know, it's a durable therapeutic, but the risk is, you know, there's off targets and there's also off tissue targets, et cetera. And, you know, I think particularly given, you know, maybe start with Laura on this one, you guys obviously have a lot of work done on this in vivo. And then also, you know, how do you think about it from an ex vivo perspective, Alex? And I think it's a really interesting area that, you know, there's been a lot of thought into. Let's start with Laura and some of the perspectives there.
Yeah, so it all starts with benefit risk, right? It starts with your indication, right? You know, then is what's the organ you're going after and the cell type in that organ. So starting with the liver, right? So, you know, using a lipid nanoparticle that where the majority of the dose goes to the liver, right, gives you, you know, first safety valve, right? So it's systemic, but is, you know, targeted. Doing good biodistribution studies, you know, informs where you need to go look for on-target, you know, activities and any potential toxicity of your drug product, which is, you know, is the lipids, you know, the other things that could go there. Then is the aspect of, you know, your editor, right, and the precision, specificity of the guide RNA and the mechanism. So, you know, they are very well established, at least for a cleavage approach, you know, that are accepted by regulatory agencies, you know, worldwide, on what you need to demonstrate. And, you know, the goal for Intellia, what we do is we put a lot of effort upfront to select guide RNAs that do not have any confirmed off targets, TJ, at multiples of the intended therapeutic dose. And, you know, that gives you, you know, safety window on, you know, you're not going to be, you know, CRISPRing unintended, you know, genes. So that is, you know, that is important. And, you know, now as part of our preclinical development, you know, we have 2 programs, you know, in the clinic, again, moving to pivotal trials with, you know, the human safety database that we're building. You know, and that really informs on the platform, right? Informs on the program, but also on the platform and leveraging that information, you know, for the next follow-on programs using the same modality, but also as you increase the complexity of the modality, whether it's an insertion or a DNA writer. So, you know, and for people developing different tools, you know, you need to be aware of what are the particular uses, the pros, but also the limitations and the concerns, okay? So there is not a particular editing modality that's free of issues. They all have different issues. You just need to know what they are and properly evaluate and control for that.
Thank you. So Alex, how do you think about it on the ex vivo side?
Yeah, it's a good question, and we have a lot of experience with bringing ex vivo products into the clinic, not just exa-cel, but we have actually 4 CAR T programs that are in the clinic now. And so this is something that we've gone through multiple times, both with the FDA and with other agencies around the world. And really, actually, I guess one thing I would point out is a lot of it is similar to what Laura said. It starts with picking a very clean guide. And I think this is different than, you know, there's publications that come out with guides that are promiscuous and you get data that looks more promiscuous that way. But if you have a really rigorous approach to selecting your guides, then that's a very important thing to start with. And then in terms of the assays for it, ex vivo is obviously going to be a little bit easier in terms of you don't have to test all these different tissue types and cell types throughout the body. And I just point out one other thing is that actually potentially counterintuitively to maybe some people in the audience is that the CRISPR-Cas9 double-strand breaks are an advantage for identifying off targets for the purposes of assuring regulatory agencies about what our off-target profile is, because the double-strand breaks serve as sort of a beacon that can then be honed in on for deeper editing around those sites, which is not as readily feasible with NICs or other approaches that don't involve cutting the DNA. So that's one area where I think the CRISPR-Cas9 technology that we're using in our ex vivo programs actually offers us an advantage that allows us to reassure regulatory authorities about what our off-target profile looks like.
And I think that is an important point. I mean, it's something, we're using, next generation editors. It's the same thing that one of the beauties of an editor that does a double strand break is it leaves a scar, a signature. You know if you have an off target, it's easily measured. That's the challenge when you move to some of the, other approaches, whether it's coupled with reverse transcriptase or a base editing approach. So Hari, how do you guys think about off targets? Because suddenly you're moving from something that today is easily measured, you know what you have, to a very different world. It'd be good to get your perspectives.
Yeah, I mean, I think we extend from similar assays. The good news is that we have a lot of assays at our disposal to understand, and you do a whole lot of this work before you even get to the clinic. And so you use analogous approaches. The good news is that we're not making a double-strand break, and so we don't have to worry about a whole bunch of other aspects that one has to worry about when you make a double-strand break. So I think it's an advantage from that perspective. But yeah, we use analogous approaches to understand on and off target, and the data suggests very favorable on target versus off target ratios.
I think it is an evolving thing outside of sort of the more established approaches which are in the clinic or hopefully commercial. That it will take time, right, to make sure we've got the right assays and analytical approaches for base editing or for rewriting or even insertions of big pieces, because it's still early. So I think that's helpful. So as we think about then, related to that's obviously regulatory, right? It's an interesting regulatory space for editing. Some of you are living this right now, and right now it appears that the strategy tends to be start in ex-US and then bring the patient data to the US. Would love to hear some comments and thoughts on that. I had the pleasure of talking with Dr. Verdun yesterday, and the way the FDA is thinking about things. But it's clearly a question all of us get all the time, I think, from folks is how do we think about getting in vivo editing approaches into the clinic successfully in the U.S. without having to approach it with patient data from other entities. So any thoughts or perspectives there? I know it's an interesting, loaded question, but we'd love to hear some perspectives. Start with you, Laura, because you guys kind of are the leading edge of the spear on this one.
Look, and I was really pleased to meet Dr. Verdun yesterday and today, and there is a sea of change, happening at the FDA. But as a new modality, right, the onus is on us to fully demonstrate that what we're bringing to the patients, fits that benefit risk, and that is going to be safe, foremost, and efficacious. So, as exa-cel being, the first, and then for us going in vivo being the first with an LNP approach, right? So you need to take the time to do everything they ask, because, they need to safeguard the safety of the patients. I think we're paving the way, as Alex was saying, to those that follow us, right? Because many of the questions are going to be answered. So, that's the thing. With regards to starting ex-US, when, we file, for ex vivo, we file INDs, for a program, what was it, in 2020, 2021. But, for in vivo, it was, we started in the UK, New Zealand. One of the advantages there were, the ability to get through your dosing cohorts much faster, which in the US was going to be very, very slow. And for us, that was a very important, consideration. Now, with regards to quality, there is, no issues, right, with the quality of other regulators, other health agencies, and that's important to note. It's not that the bar is lower there. We now have multiple geographies where we have open CTAs, and the quality of the review, the questions, is superb.
That's great. And I do think, I agree with you fully. Dr. Verdun was fantastic. Like, the thinking and the ability to enact change there is going to be really wonderful to see over the coming years. And so, Alex, how are you guys thinking about the in vivo and sort of how you think about going in the clinic and where? And then, Hari, if you can follow up.
Yeah, well, obviously we're looking closely at what happens with Intellia. That's probably they're the leading company in this space, and so we get to learn from everything that's been happening, and I think they're making great progress, and that benefits everyone. One point you made is that there's a trend to go ex-US and then move into the US, and I think that's fine, right? There's a lot of drug development programs, not just in gene editing, but just historically across the field that go ex-US for various reasons. Europe historically has been a little bit faster to get a first-in-human trial started, so that's not unusual. Going to Australia and New Zealand is fairly common. So the fact that companies are going ex-US and then moving into the US isn't inherently problematic. I think what we need to see is then being able to make that step of, for later stage studies, certainly getting into the US, and we've seen now that Intellia cleared an IND for HAE, which is definitely great progress.
Hari?
Yeah, I mean, we second Alex's comments. I think we'll learn from people who've come before us, and look at the kind of optimization of the overall clinical investigation, whether it is the regulatory pathway or the operational pathway, and look at it holistically and see which is the most optimal way to get this into patients. And so it could involve ex-US pathways, but in other cases it could involve US first pathways.
Yeah, I think for companies like ours that are looking to go in the clinic in the next period of time, it's wonderful to see someone breaking that wall down, and thank you for that. It helps the entire space. So we just have a couple minutes left. It'd be great to hear your perspectives 5 years from now, where I think we'll be in Arizona at the MESA meeting in Arizona in 2028. What does Intellia look like? What's Tessera look like? What does CRISPR look like as a company? So let's start with Tessera, and then we can go to Laura, and then over to Alex.
Somebody by the name of Yogi Berra said it's difficult to make predictions, especially about the future. I think 5 years is a long time. By then, we hope to have multiple products in the clinic for sure, and validate our technology, liver, but also extrahepatic tissues as well. And the beauty of the kind of medicines we're all developing is the path to late stage and commercialization is fairly streamlined once you get past that initial phase. And so hopefully we'll be staring at some of those endpoints in that time frame.
Yeah, for me, look, I think we're going to have important medicines, that are going to be impacting people's lives positively, that are going to be reimbursed, and that they're going to be, leading to this change in how we do medicine, right? And, looking at, potential cures rather than treating diseases, changing the lives of patients with cures.
And for us, obviously, first and foremost with exa-cel, our hope is that in 5 years we'll have a commercial product there, and not just a commercial one, but one that has robust sales. I think there's still a big question mark in the field of what's the commercial potential of especially ex vivo gene editing, and so our hope is that the sales for sickle cell and beta thalassemia are going to be robust. We think they will. And then beyond that, looking at the rest of our portfolio, we expect that in 5 years we'll have multiple in vivo editing programs in the clinic, both hepatocyte-directed and outside of hepatocytes. We have our CAR T programs that by then we expect to have pivotal data in both lymphoma and potentially having data by then in solid tumors with our CTX131. And then lastly, our diabetes franchise, where we have VCTX211 that is now dosing patients. Our hope is that we'll have learned from that, and we'll have diabetes programs that are producing meaningful amounts of insulin and better controlling people's glucose.
Great. Well, we'll see you guys in 2028. So last point I think is important, that we do work together actually under the auspices of ARM. So the draft guidance on gene editing, actually, all the editing companies got together and we worked together to have a joint view of what we thought was appropriate from that. And so I think it is important to note that while we are competitive, we actually do work together on important common things, and that's usually on the auspices of ARM. So again, thank you all for joining, and thank you panelists. It was wonderful to have you here.
Thank you.
Thank you.